testify on the bill. And we are also joined by Delegates Hartman, Vogel, and Wells. And the title of the bill is Animal Testing and Research, Human Relevant Research Funding, and Animal Testing and Research Licensure. It is jointly assigned to us in Ways and Means. I want to welcome Delegate Hartman. And if you have any questions when we come to that portion, will you let me know? You're welcome to sit here if you like, okay? Thank you. So we're going to have, if I may have the following panel. I have Kathleen Conley.
Yes.
Eric Galley, Keith Murphy, and Jim Corbett. Will you be so kind and come up, and you can pull a chair. Brigida, would you like to get started?
Yes. Hi. My name is Brigida Kristofic. I am the Chief of Staff for Delegate Cullison. Thank you for having me here today. Thank you for this opportunity to present HB 626, which is the Animal Testing and Research, Human Relevant Research Funding, and Animal Testing and Research Licensure. Ooh, a mouthful. This bill creates a fund to spur the development of human relevant research, driving the development of emerging research technologies that do not use animals. The fund is modeled after the Stem Cell Research Fund administered by TEDCO and includes a license fee on institutions that engage in animal testing. Maryland should be proud of its work on two fronts: to invest in its biotech industries and research institutions, and to create strong animal welfare protections. Specifically, I am referencing the Center for Alternatives to Animal Testing at JHU, at Johns Hopkins University, and the Emerging Biotech Center at University of Maryland. HB 626 will ensure that Maryland stays a leader as new non-animal testing methods move to the forefront. We're already seeing the impact that human-relevant technologies have on advancing human health and discovering cutting-edge solutions that animal testing could never have revealed. In fact, testing on animals is not even necessarily as accurate. 90% of drugs found safe on animals ultimately fail in human trials. Investing in these emerging technologies will take Maryland to the next level in our research, our biotech, and our life-saving interventions. Maryland has already established that we want to be a humane state. Legislature in 2021 passed a law ending the testing of new cosmetics on animals. The removal of 4,000 dogs last summer from a center that bred for research shone a spotlight on the inhumane treatment animals in research too often face. While those 4,000 dogs were able to start new lives in loving homes, more than 60,000 dogs are used in laboratory testing each year across the country. And we can move away from animal testing by investing in these innovative research that is humane. We are working with stakeholders on amendments as to the funding mechanism and to have institutions that test on covered animals, animals under the protection of the Federal Animal Welfare Act, to make contributions to the fund. That would not require new reporting or oversight, as those institutions already must receive USDA licenses and report the number of animals they test on. It would also strike the penalty provisions on the bill. This bill is a revenue-neutral way to move Maryland towards the future we want, one with solutions to the hardest medical questions, and one that is less reliant on outdated animal testing. There is broad stakeholder agreement with the amendments, and I respectfully request your favorable consideration. And with that, I will turn it over to the rest of my panel. Thank you.
Wonderful. Who will be next? And we've been joined, if I may, by the other members of the Ways and Means Committee, Delegate Vogel and Delegate Wells. Welcome. If there are any questions, please let me know, okay? You may get started.
Thank you, Madam Chair, Madam Vice Chair, also our sponsor, members of the committee. My name is Eric Galley, and I'm here today representing the Humane Society of the United States. I want to thank the sponsor, also the Vice Chair, for all the hard work she has done on the bill. I also want to thank Delegate Rosenberg. His stem cell research law was the model we used when drafting this legislation. Thank you for paving the way. You might say we stole his language. Depends on how you feel about it. He's been very generous about it, I have to say, so I think he's okay with it. We also want to thank our friends at the University of Maryland and Johns Hopkins University. They have worked diligently with us to make a deal on amendments that leaves them comfortable with the bill and leaves the basic principle intact, that we will do this important research and that those who currently use animals in testing will help finance it. I wanted to briefly review the amendments that the Humane Society, Hopkins, and the University of Maryland have agreed to. We have not had time to fully write them up yet. They're extensive, but the concepts are pretty clear. You will also find these concepts in the written testimony from Michael Huber of Hopkins. And those briefly, those items are: we're going to switch to the definition of covered animal in the Federal Animal Welfare Act, which is a big change. We were originally going to cover more animals. We're going to drop the licensing and fee structure, which will help significantly with the fiscal note. Wasn't the original reason, but it helps. And the licensing and fee structure will be replaced by a mandatory contribution for each entity based on how many covered animals are held that year. And the criminal penalties will be removed. So those are the fundamentals of the amendments, and we're very glad that they've worked with us, and we appreciate everyone's help very much. Thank you very much, and I'm happy to answer any questions about the bill. Thank you.
You're welcome, and thank you for working it with the advocates. Kathleen Conley.
Yes, thank you, Chair, Vice Chair, members of the committee. My name is Kathleen Conley. I'm Vice President of Animal Research Issues with the Humane Society of the United States and reside in Frederick, Maryland. I actually used to work for several years at a primate breeding and research facility, and I went in thinking I wanted safe and effective treatments for humans, and I left because I knew we could be doing better than we were for both humans and animals. So that's why I'm really excited about this bill today. The world is moving forward toward a future dominated by using human cells, 3D printing, computer modeling, and other technologies that do not rely on animals and only continue to get more sophisticated. You'll be hearing more about these from others on the panel. Animal experiments, on the other hand, will always have severe limitations. A mouse will always be a mouse, a dog always a dog. They will never be human. Animals have substances forced down their throats, into their lungs, in their eyes, on their skin, and they suffer for months or years before being killed. And we heard drug failure rates are extremely high, largely because we're taking misleading results from animals. So this has caused hospitalizations and even deaths. Government agencies are moving away from using animals and investing in these technologies, and really Maryland can get out in front as an investor in these. So as HB 626 establishes a human relevant research fund through an annual contribution, as we heard, by institutions using animals covered by the Animal Welfare Act. We appreciate the willingness of the institutions and engaging with us on possible amendments. And finally, really the public cares about this a lot. According to a poll earlier this month, Maryland voters, 79% of them favor investing in these technologies. So Maryland really could become a leader in 21st century science and increase human health and safety while also protecting animals. Therefore, we urge a favorable report on HB 626.
Who's
Thank
next?
you.
Thank you for your testimony.
I wasn't sure if I was called up
What
to
is
this
your-
panel
what is
yet.
your
Aisha
name?
Akhtar.
You can go ahead.
Okay, all right. Thank you, Madam Chairperson, and members of the House Committee. Thank you for giving me this opportunity to present today in favor of HB 626. I speak to you on behalf of the organization that I have co-founded and am president and CEO, which is the Center for Contemporary Sciences, which is a nonprofit health organization based in Maryland. And I speak to you as a personal citizen and a neurologist and a public health specialist. One of the hardest things I have ever had to do as a neurologist is to watch my own aunt, a strong, incredibly vibrant human being, deteriorate from Parkinson's disease until she died. I watched helplessly as she slowly lost control of her arms, and a truly terrifying experience, as you can imagine, losing control of your own body's movements. Her arms pained continuously from the constant, uncontrollable tremors. Meanwhile, her legs refused to move increasingly until the end when she was unable to walk, to stand, and perform the most basic bodily movements that we take for granted. I tell you my aunt's story because there is not a single effective treatment for Parkinson's disease, nor is there an effective treatment for multiple sclerosis, dementias, spinal cord injury, most cases of stroke, and just about every neurological disease out there. And as a neurologist, having to tell and diagnose patients with devastating neurological diseases, it's incredibly difficult to tell them, I'm sorry, but there is no effective treatment for your actual disease, nothing that will change the course of your illness. In fact, there is no approved medical treatment for most human illnesses out there. And as you've heard, one of the large reasons that has come about in recent decades, and what I learned as a medical officer in the Food and Drug Administration, is that whatever role animal testing may have played in the past, it is now largely misleading results and causing the potential abandonment of cures and effective treatments. So I urge you to please vote in favor of this bill, help Maryland become a leader in exploring new ways of medical research, ways that are actually based on human biology, thus they are human relevant, they are kinder, they're more effective, and perhaps more patients like my aunt and future patients with neurological diseases won't have such devastating diseases to endure. Thank you.
Thank you. Mr. Murphy, can you please unmute yourself and give us your testimony?
Thank you, Madam Chair and Madam Vice Chair, for the opportunity to submit testimony on HB 626, critical legislation that would advance the use of non-animal testing methods in medical and product research and mark Maryland's commitment to developing safer and more successful drug therapies. I'm a biotech entrepreneur based in Southern California. I and those in my field appreciate your efforts, and I wanted to provide the perspective of an entrepreneur and innovator working on new non-animal testing methods. I am here to tell you that we are at the cusp of a major technological revolution. That technology exists today to dramatically improve our ability to develop drugs, improve the likelihood of success of drugs in clinical trials, and lower the costs of drug development and thus drugs themselves. For patients, this can mean more new breakthrough therapies and lower priced drugs. But first, the technology needs to be taken up more broadly and more solutions need to be developed, which HB 626 grants will support. Several companies now are in the early stages of applying more modern technologies, such as 3D bioprinted tissues made from human cells from a patient, to avoid the species gap and over-reliance on rodent and animal models in pharmaceutical research. I am the Chief Executive Officer of Vizient Biosciences in San Diego, which has a superior model for liver fibrosis made from 3D human cells. Several major big pharma drug programs in this space have failed to reduce fibrosis in patients, and 2 have been successful. Animal models predicted all of them would work, but our 3D human model shows exactly which 2 reduce fibrosis and which ones fail to do so. These models and other technology to do better than animal models exist today and need support to be more broadly used. Our company is one of several, some of which commercially offer their models and their solutions to drug developers. Your efforts through this legislation would yield great benefit in making these models be used sooner and with greater impact. The result will not only be beneficial in the sense of less use of animal models, but will end the ethical tragedy that is using patients in experiments doomed to fail when the technology exists to avoid such practices.
Thank you for being with us. Mr. Corbett.
Thank you, Madam Chair and members of the Maryland House. My name is Jim Corbett. I'm the CEO of Emulate, a leading organ-on-chip technology developer, and I offer my support of House Bill 626. There's no doubt that animal models have contributed to major scientific advancements and to safe and effective drugs making it to market. However, these models have a difficult job of approximating the human body, and sometimes they get it wrong. Over the last 12 months alone, there are 6 high-profile examples involving well-known companies that saw drug-induced liver injury present a problem, either in terms of stopping clinical trials or, in one case, causing fatalities, after passing through animal testing requirements. 21st century science is proving there's a more safe and more humane path forward. In a recent study published in Communications Medicine, part of the Nature portfolio, researchers found that Emulate human liver chip to have this 87% sensitivity and 100% specificity when differentiating hepatic toxic from non-hepatic toxic small molecules. Importantly, all 22 of these hepatic toxic drugs in the study had previously been classified as safe due to a lack of toxicity in animal models. Collectively, these 22 compounds resulted in 208 patient fatalities and 10 liver transplants. Had the human liver chip been used during the preclinical screening of these compounds, it's likely that many of these fatalities could have been avoided. Moreover, researchers also found that improving our ability to detect hepatotoxicity with 87% sensitivity, it's estimated that the human liver chip can increase research and development productivity by $3 billion for the pharmaceutical industry on an annual basis. However, transitioning to human-relevant research testing methods requires funding programs in order to continue to spur scientific innovation. Funding programs for human-relevant research will lead to new education and career opportunities, as well as positive impact on the economy of Maryland. For these reasons and more, we urge the passing of House Bill 626. Thank you.
Thank you for your testimony. This is the first panel. I'm going to open it up for questions now for both committees, the Ways and Means and the Health and Government Operations Committee. Since there are guests, we're going to start with the Ways and Means Committee. Are there any questions? Seeing none, the Health and Government Operations Committee, we have Delegate Shalega.
Thank you, Madam Chair. I almost called you Madam Speaker again.
I don't want her job. I'm happy where I am.
I feel better at least making that mistake here than on the floor. Can you tell me, is this going to expand embryonic stem cell research, or are there embryonic, parts involved in this bill?
No, this does not at all touch on embryonics research. This is using human stem cells from adult stem cells, often, or other types of human tissues that have been ethically derived, voluntarily derived, or discarded from surgical cases and so on. These are these are the tissues that make up these types of testing methods.
No embryonic.
That is correct.
Okay. I have Delegate Kipke.
Thank you, Madam Chairman. A quick follow-up to that question. Is there some information that you could provide in writing related to that so that we have something to back that up?
You
Yes,
can give it to us offline. Can you do that, please?
Yes, we can absolutely do that.
All right. Will you be so kind and send it to the delegate and the entire committee, okay?
Absolutely, yes.
And then to the virtual people, either Jim or, I can't read your name, Keith, I believe.
Yes.
Why is a government solution to this necessary? Why isn't industry moving in this direction without the help of government? Whoever is best equipped to answer the question. I assume both, at least one of the companies here is going to profit off of this. So I know why you might be interested, but can you tell me why it's necessary to have the government involved in this?
I'll jump in because my company will not profit off anything coming from your support of this bill. We don't sell any solutions. We're only developing our own drugs using our interior technologies, but I see a public policy benefit broadly for this because it's not going to move fast enough. Pharma scientists have been trained in the use of animal models. It takes them a long time to come to an understanding of how they should be used. There are very slow processes at work that will slowly turn over, but it's going to take a very long time. This is an opportunity to accelerate the breadth of the penetration of this throughout, a broad set of uses, and Maryland researchers will be developing some of those technologies. They'll be first used in Maryland. So I think it's the speed with which we can benefit things akin to supporting any new technology with tax breaks or other things. This really allows it to, reduce drug prices. I think that's one of the biggest impacts. Create new drugs that won't exist otherwise faster and, reduce the cost of drugs sooner for Maryland residents.
Delegate Riley.
Thank you, Madam Chair. Yes, so I've heard a lot of testimony on the conducting medical testing, but we also have in the bill that it also addresses the product testing. Can you give me some examples of products?
Cosmetics, drugs, pesticides.
All right, thank you.
Any other questions from anyone? Seeing none, thank you very much for your testimony and for your work and your time.
Thank you.
Next, we're going to the
Thank you.
second panel. My pleasure. We have 4 virtual testimony. Will you be so kind and allow them all to come in, please? And for the witnesses that are waiting, thank you for being patient. For the committee also, thank you for your time and your patience. Ms. Leary, will you be so kind and get us started while the rest of the panel comes on? How are you today?
I'm fine, thank you. It's all been very interesting.
That are waiting, thank you. Can you turn off the, yeah, then you can get started.
Just did. Thank you.
Thank you.
You bet. Well, good afternoon, and thank you, Madam Chair, Madam Vice Chair, members of this committee for providing this opportunity. We are testifying in support of HB 626. My name is Sue Leary, and I'm president of the Alternatives Research and Development Foundation. We've made grants totaling over $4 million to advance new approach research methods that do not use animals. These methods, as you've heard, are model systems based on human biology and comprise some of the really most exciting and innovative research and testing and biomedical research. So I have submitted written testimony. I ask that you take a look at that because it has more extensive comments and references. But today I did want to highlight that our nonprofit foundation has proudly supported a number of projects at highly respected research facilities in Maryland, which, as you know, has a unique concentration of scientific expertise. But I want to emphasize that we are well aware that more investment is needed. Our foundation can only do so much. We enthusiastically support this legislation's plan to establish a fund for human relevant non-animal testing and research. These methods show enormous promise to really break through and rapidly advance human health and reduce animal suffering, but a considerable investment is needed. The fact is that our experience has shown that incentives work and funding does motivate innovation. So as has been mentioned, some of these very exciting systems have been developed recent years, astounding progress developing these technologies, and this is recognized, and anyway, we support a favorable report on this bill. Thank you very much.
Thank you, Ms. Leary. Mr. Hickman?
Hello, thank you very much for the committee for allowing me to speak. I'm in support of this bill. And I just wanted to say is that I've been working in this area for about 30 years. And one of the founders, and I founded a company with Mike Schuler, who is considered the founder of the field, about 7 years ago. And we have been working with commercial entities to be able to expand the breadth of knowledge in this field, and also to try to educate people. And that's partially what I'm trying to do for this committee. And that the main thing is to try to get regulatory acceptance of these models in live animals. And it has to be understood that there's a big difference between looking at efficacy, does the drug work, versus looking at safety. And the critics of the bill are going to say that, well, you're going to always need animals, and they're correct for safety, okay? But not necessarily for efficacy, okay? And this is where we're at right now, where we're already developing models for efficacy. Our company had helped Sanofi to get a drug repurposed, okay, that had safety data in humans into a clinical trial with just efficacy data from one of our models. So the regulatory folks are actually in favor of this. And also, lastly, speaking for the chance of the rare disease community, is everybody says, well, either animals or these new microphysiological systems. But actually, for rare diseases, there's over 7,000 rare diseases, only 400 active research programs. So not only will you be helping diminish the number of animals, you'll also be helping a whole class of folks who don't have animal models to rely upon if you actually increase funding in this area in this bill. Thank you much for your time.
Thank you for your testimony. Mr. Armento.
Thank you, Madam Chair, and good afternoon to the honorable members of this committee. My name is Alex Armento, and I'm the president and CEO of MatTek Life Sciences, a Massachusetts-based biotechnology company and a global leader in the development and manufacture of human-relevant alternatives to animal testing for product safety and efficacy. We are tissue engineers, and we use human cells to create lab-grown, miniaturized human tissues which mimic the structures and functions of native human tissues. To date, we manufacture 15 different types of human organ tissues, both healthy and diseased, including skin, eye, oral, nasal, lung, intestinal, and vaginal tissue. These models have been cited in more than 1,000 scientific publications and more than 500 patent citations. In addition, 6 of our test methods, including skin irritation and eye irritation testing, have been fully validated by regulators as standalone alternatives to animal tests, such as the rabbit Draize test. We conservatively estimate that the use of our human-relevant tissue models by our customers in the pharmaceutical, personal care, and cosmetic industries have saved the lives of roughly 100,000 laboratory animals in 2022. Even more important than the ethical goal of sparing laboratory animals, the companies that are tasked with developing safe and effective consumer products and pharmaceutical therapies can obtain testing results that are much more relevant to actual human biological responses. My last point, MatTek is also a success story as it relates to government funding. Over the first 25 years of our existence, we were awarded several SBIR Phase 1 and Phase 2 grants to develop many of our existing tissue models and test methods to predict tissue-specific human toxicities. Although we no longer seek these types of government grants, MatTek has built a growing and sustainable business that will continue to lead in the field of human-relevant testing. We now employ 75 people across scientific and administrative positions, and as we continue to grow our business, we'll always be grateful for the previous government funding that still allows us to have an impact in the field today. On behalf of our team at MatTek Life Sciences, I strongly support HB0626. Thank you for your time.
Thank you for your work, all of you, and your time. Ms. Fullerton.
Good afternoon. My name is Kim Fullerton. I'm Vice Chair of the American Bar Association Animal Law Committee, and I'm here today representing the Maryland State Bar Association Animal Law Section. I've been briefed on the amendments, and I do find them to be appropriate. And it's also important to note that with these amendments, the bill will be very narrowly tailored. So it will serve solely as a funding mechanism. It does not require new reporting or additional oversight. The penalty provision will be stricken, and the regulatory component is a completely separate bill. So the only issue that is presented to this committee in passing this bill is, as the delegate mentioned earlier, should Maryland promote state-funded medical research to develop human-relevant testing methods? And that answer is yes. It would benefit so many of the people and issues that we've heard about earlier today. And also, we have some of the best research institutions in the country. This bill would directly benefit them as well. This bill is how we accomplish those goals. The funding mechanism aims to be revenue neutral, and it will not depend on government funds to achieve our goals. The framework is modeled after another research fund, which is already administered by TEDCO. So we have high confidence that this bill is an effective strategy for driving critical research and more effective testing. To sum up, Maryland is already a hub for biotech and research. This bill is how we will preserve that interest while incentivizing research that benefits our state, our people, and our animals. Thank you.
Your testimony was excellent, very clear. Thank you. Are there any questions for these 4 virtual witnesses? First, Ways and Means.
Wish I did my first time in the front row, probably my last.
Well, you're welcome in HCO anytime. Okay, so we do have Delegate Martinez.
Thank you, Madam Chair. Could someone on the panel, or just tell me how many organizations, foundations, or research institutions in Maryland currently conduct human-relevant research?
Do any of the 4 panelists, if you know, will you unmute yourself? If you don't, it's okay. We can ask that question offline.
I can provide you more detailed information offline.
Thank you so much. Can you send it to Delegate Martinez and the entire committee as well, okay?
Absolutely.
Thank you very much. We have Delegate Hill.
Thank you, Madam Chair, and thank you all for this, for being here and for this important work. Simple question. There's a lot of federal funding that's going into this same thing, National Cancer Institute, even the USDA. Why does Maryland need to provide additional grant and underwriting for this kind of research when the federal government and private institutions are already doing it?
Who will be the best person to answer? Okay, Mr. Hickman, you raise your hand. Go ahead.
Yes, the reason why was the one gentleman earlier said it best, is that to accelerate it. Okay, there is some investment in this field, but compared to the overall research endeavor out there in terms of pharmaceutical industries, animal models and things, and this is only a very, very small fraction devoted to microphysiological systems, human-on-a-chip-based systems. All we're looking for is to increase that and be getting any help, federal government, state governments, okay, as well as commercial funding for this area will help promote this faster, quicker, okay, and be able to alleviate suffering for patients who just don't have any other hope of using traditional methods.
Thank you. Any other questions from the committees? Seeing none, thank you very much.